So, Assalamualaikum and hi. Today, my group and I, which is group 2 from A4AS1203_7, we have chosen this topic, which is immune system, for our video presentation. So, without further ado, let's go. So, before we get into the topic, I would like to introduce my team members. So, first we have Amira Huda binti Aswa, Nizamaisara binti Abdul Hamid, and we have Muhammad Danish Imran bin Mus Mulyadi, myself, Nuriza Karina binti Kamarzani, and besides me, we have Priya Irina binti Muhammad Hadir. So, what is immune system? Okay, according to Oxford study, immune system is the organs and processes of the body that provide resistance to infection and toxins. The immune system keeps a record of every microbe it has ever defeated, so it can recognize and destroy the germ quickly if it enters the body again. There are two major stages of immune system, which is innate immunity and adaptive immunity. The innate immune system consists of two lines of defense, while the adaptive immunity, also known as acquired immune system, has two different mechanisms. Active immunity and passive immunity are two types of acquired immunity. So as I've mentioned earlier, innate immunity has two lines of defenses. I will be talking about the first line of defenses. First and foremost, we have intact skin. It provides a physical barrier and it is tightly packed with keratinocytes. It prevents direct entry of microbes. However, abrasion, wound, or bites may allow their passage. Secondly, we have mucous membrane. It acts as a physical barrier against pathogens. It secretes a sticky and thick fluid called mucus, and this mucus works by trapping debris and particulate matter, including microbes, from going through the underlying cells. So last but not least, we have secretion. First, we have from skin. So what does skin secrete? Oil and sweat. Why? To maintain low pH, flush out microbes, and support skin barrier's function. Next, we have stomach. What does stomach secrete? Stomach acid. Why? To kill harmful bacteria or pathogens that are ingested with food. And last but not least, we have tears and saliva. Tears and saliva both contain lysozyme to kill bacteria by breaking down their cell walls and prevent them from attaching to mucosal surface.
Next, I'm going to talk about the innate immunity, which is the second line of defenses. So first, we have a cellular innate defense, which is the phagocytic cell. So from the left picture, we can see it is a process of phagocytosis. So we have 6 steps. So first, the pseudopodia will be surrounded the bacterium, and the bacterium are engulfed into the cell. So vacuole that containing the bacteriums are formed. So vacuole and a lysosome will be fused. So toxic compound and the lysosomal enzyme destroy the bacterium. And then lastly, bacterium debris is released by exocytosis. Then we move to the right picture, which is it is talk about the phagocytic cell and what is the function. So first is a macrophage. It will be in gas and destroy the invader. And the second, we have a neutrophil, which is the engulfing and destroying the cellular debris or invading the bacteria. Third, we have a monocyte, which is it will be in gas and destroy invaders. And fourth, we have an eosinophil, which is it defends against pathogens that have been coated with antibody. And lastly, we have a dendritic cell, which is it will stimulate the development of acquired immunity. Second, cellular innate defense, which is natural killer cell, or we call as NK cell. So it can patrol the body and attack virus, infected body cell, and cancer cell. Then it will be trigger the apoptosis. So what is apoptosis? So apoptosis is a physiological process in which cell death occurs through a regulated sequence of events. It leads to the programmed elimination of specific cells without causing damage to the surrounding tissue. Third is inflammatory response. So when our tissue injury, so the chemical signal will be released, which is the histamine. And second, dilation and increase the leakiness of capillary. So phagocyte will be migrate to the area. And lastly, the phagocyte will consume the bacteria and the cell debris. So platelet move out of the capillary to seal the wounded area. Lastly is the antimicrobial protein. So it consists of two, which is the complement system, and second is the interferon. So we are going to look at the difference between them. So we are going to look at the complement system first. So the effect is it will be activate the phagocyte, it will trigger the inflammations, and lastly pore formation. For interferons, so it will be activate the macrophage and the NK cell. Second, it will be induced nearby an infected cell to produce the proteins that inhibit the viral replication.
Humoral immunity is a type of acquired or adaptive immunity that specifically involves B lymphocytes. It operates mainly in body fluids such as blood and lymph, which is why it is called humoral. Each B cell has a unique receptor on its surface known as B cell receptor. When an antigen enters the body, only the B cell with the matching receptor can bind to it. This specificity ensures that the immune response targets only the correct pathogen. After the antigen binds to the B cell becomes activated with help from helper T cell. The helper T cells release cytokines that stimulate the B cell to divide rapidly. This process is known as clonal expansion, where many identical B cells are produced. Some of these B cells differentiate into plasma cells, which actively secrete large amount of antibodies that are specific to the antigen. Not all activated B cells become plasma cells. Some differentiate into memory B cells, which remain in the body for a long time. If the same pathogen enters the body again, these memory B cells allow a much faster and stronger immune response. Antibodies produced during this response neutralize toxins and viruses, promote phagocytosis, and activate the complement system. This mechanism is also the basis of vaccination and long-term immunity.
So, we just saw how antibodies fight viruses outside the cell. But what happens when the virus hides inside our cells where antibodies can't reach? That is where my part comes in, cell-mediated immunity. The system relies on T lymphocytes or T cells. Unlike B cells, T cells are designed to attack infected body cells, cancer cells, or foreign tissues. They start their life in the red bone marrow, but they migrate to the thymus gland to mature, which is why they are called T cells. There, they are tested to make sure they don't attack our own healthy body. Now, how does a T cell fight? It starts with a process called clonal selection. Imagine a T cell as a soldier looking for a specific enemy. When it finds and infects a cell that matches its specific receptor, it gets activated. Once activated, it doesn't fight alone. It starts dividing rapidly, making thousands of copies of itself. This is called proliferation. Basically, one single cell into a massive army specifically designed to destroy that one type of virus. This army then splits into three specialized teams. First, the helper T cells. These are the commanders. They release chemicals to help the B cells and macrophages fight better. Second, the cytotoxic T cells. These are the killers. They touch infected cells and release toxins to destroy them instantly. And finally, the memory T cells. This guy don't die after the battle. They stay in our blood for years. If the same virus come back, they remember it and kill it before we even feel sick. This memory is the basis of how we become immune. But how do we get this memory without getting sick? First, that is where the vaccine comes in. And my next person will explain how the active and passive immunity works. Thank you.
We have two types of immunization, which is active and passive. Active immunity is a natural exposure to infectious agent, and it is an artificial immunization. It will develop naturally in response to an infection, and it has own antibody. For example, immunization when vaccination, because a non-pathogenic form of a microbe and immune will respond to an immunological memory of microbe. Passive immunity is a natural maternal antibody, and it is an artificial antibody that we get from the other sources. It provides intermediate and short-term protection. It is more faster than active immunity. It also has ready-made antibody too. It will convert naturally when two process, which is IgG crosses the placenta from mother to fetus, and the second, which is IgG passed from the mother to infant that located in the breast milk. And it also conferred artificially when injection of the antibody.